Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi, everyone. Welcome to the holiday special of the research roundup that we've been doing every quarter this year with Dr. Veera Rajagopal, who's here today. For those of you who haven't heard this kind of episode before, and especially in the holidays, we tend to go a little bit longer. So this is going to be probably more than an hour of some of the latest interesting research in the last quarter. We're going to cover hopefully four different papers and stories today that span a pretty wide range, from some population genomics programs, newborn screening, and some kind of fundamental mechanistic research in a way in Alzheimer's. So we're going to get into it in a second. But Veera, for people who haven't come across you on Twitter or on here before, I'd love if you could just give people a quick intro to your background, how you got into this amazing side hobby that you have of breaking down the newest research every day sometimes. That your output is prolific. I find it amazing.
Thanks. Thank you, Patrick, for inviting me for the podcast. Yeah, so I'm Veera Rajagopal. I'm a research scientist at the Regeneron Genetics Center. So I'm a physician by training and been working in the field of psychiatric genetics for quite some time. And at the Regeneron, I'm focusing on translational, using human genetics data to discover drug targets and also support drug development by bringing in genetic insights, some large-scale genetic data, focusing on mostly neurological diseases, particularly psychiatric and addiction-related diseases. So yeah, I do spend quite a lot of time in Twitter. Maybe not so much recently, but I've been very prolific at least 2 years ago. And yeah, I do read a lot of papers and try to summarize them. I use Twitter like a kind of a notes where I just, whenever I come across some interesting findings, I kind of quickly record it in the Twitter, and that really helped a lot of people to follow the literature. And that's how it started. It's trying to keep up, but it's a challenging time. And also the Substack thing I've been doing this past 1 year, trying to write long form posts of most interesting papers that, that I wanted to read fully and understand more dive deep into the background. And most of our previous podcast discussions are focused on such works. Yeah.
Yeah, absolutely. We'll put links to the Substack as well as Twitter in the show notes for anybody who wants to follow. A lot of these will either be on there in the future or already on there. So if you listen to any of our quarterly roundups and you prefer to read or you want to read through after listening, then you can always go find them on Veera's Substack. So let's go into the first paper. You picked this one. The title of it is Insights into the Causes and Consequences of DNA Repeat Expansions from 700,000 Biobank Participants. Why did you pick it, and what's interesting about it to you?
Yeah, so I've been like I said, I've been studying a lot of papers and going through the literature in a broader aspect, right, of the human genetics. And always, sometimes I start getting interested in a particular gene or, like a finding, or sometimes a particular trait or disease or a technology, right? So one of the two papers that I will be discussing today is one set comes under that category. So one particular topic that really intrigued me is the repeat expansion, especially after, the recent really fascinating work that came out of Steve McCrull's group on the repeat expansion, Huntington's gene repeat expansion, triplet repeat expansion in the striatal neurons, right? So was one of the top talks in the ASAG 2 years ago. And we had a preprint this year. And there is so much also going on in therapeutic space. So I'm starting to learn more on the repeat expansion. And there is also the whole genome sequencing, large-scale data sets picking up. And we are learning a lot of repeat associated diseases and, so recently. And so that is why this is kind of very interesting, because this is it comes under the lot of topics that we already discussed. We will see that, in detail. And the other one is the genetics of age at menopause, particularly the female reproductive traits. That's like really interesting to me. Particularly, we are learning a lot on this based on the UK Biobank data. We will come to that later, but focusing on our first paper, the repeat expansion here. So this is a paper. It's a preprint. It's actually from the same group of authors we keep discussing almost every podcast episode. The Laws team at the Harvard and also Steve McCrow's group. So this is like in here, what they're doing is they are just using the newly generated whole genome data in the whole genome sequencing data in the UK Biobank and also in the all of us cohort. And they're focusing here on a particular kind of mutation called short tandem repeats. So particularly focusing on the CAG triplet repeat. It's the same repeat that we actually discussed in the context of Huntington's. And here, what they are actually looking at it is so they are focusing only on the CAG triplet repeats. And so they actually, it's very difficult to actually identify repeat sequences from short read whole genome data, right? So because the maximum length of each read is like 151 base pairs. So you are really limited. So they are using a lot of really, intelligent way of looking at it. So they specifically focusing on the CAG repeat, and they are restricting themselves to the repeat, repeat lengths of like probably greater than 45 repeat units, which is what, you can accommodate within and 151 base pair reads. So basically they start with a very simple question like, okay, let's look at the genome wide, across the human genome, all the regions where you would find really expanded more than 45 repeat units of CAG triplet repeats in the genome, And when they do that, they actually are able to identify at least 18 sites, genes where they were able to identify at least 5 participants among the 700,000 carried an expanded repeat greater than 45. And so among these 18 sites, there are 3 sites that contributes more than 97% of this repeat expansion. That's like carbonic anhydrase 10 gene, and then there's a TCF4 transcription factor gene, and then ataxin ATXN gene. So these 3 sites alone, like there are thousands of participants. Like I think like for the carbonic anhydrase, there are like nearly more than 130,000 individuals who carry more than 45 repeat units.
These 3 are already associated with a number of diseases, right? They discovered 2 novel ones, but these ones are quite well known. TCF4, for example, is I think the repeat expansion results in the Fuchs endocorneal eye disease. So these are kind of well understood repeat expansions, right? The ones that they found here.
In some sense, right? So like that's one of the highlights is like all our knowledge of this, repeat expansion diseases, and also later we will see for the primary ovarian insufficiency. All this comes primarily from isolated cases from Mendelian literature, right? So it's really, like our understanding of the penetrance and, like the prevalence of this repeat expansion at the population scale is very, very limited because we don't have the data set yet. And right now we are generating data in a very large scale. And so a lot of insights we get from this is really interesting. So, and of course, like, because of the limitations, you will not be able to go and find a new repeat gene, right? Because that would mean that you are doing a discovery. For that, you would really need a very, like a precise way of estimating the repeat, which is not possible using short read whole genome sequencing. And that is the main reason they just go into the very well known, understood, like the triplet repeats, which they can estimate and they can validate as well. Yeah, but what you say is correct. So we know about the triplet repeats in these genes, but there are more insights that's coming up with the data here. They are looking at it. And so it's really, like they mainly focus on 2 aspects. We can divide this into 2. One is like the germline repeats, right? So the germline repeat, and then they also focus on the somatic repeat expansion. In the germline repeat expansion, like, in the past discussions, we mentioned about haplotype based methods, that many haplotype based imputation methods that Purlow's team have developed, right? Using that, they were able to identify coding variants even before exome sequencing data was released. And also they were able to identify variable number tandem repeats, which is the other category of, short repeats. And we have seen a lot of new disease associations. A lot of them were underlying, were the driving variant in the GWAS locus, right? So similarly here, so they are looking at this, like this tandem repeats. And so one thing they're looking at it is like, like the mutation rate. And so for that, they use this, like they kind of use this creative way of using the identity by descent analysis. So typically for, like estimating the mutation rate, what you would need is like multiple generations genome data from, like multiple generations, right? That's why typically
Right.
these estimates come from data sets like decode, right? So who actually, from which we have learned about the genome-wide SNV, like mutation rate, something like 70 to 90 mutations per, and then also there are estimates for the short tandem repeats, considering around 1 million STRs prevalent across the genome. You get like, so based on that, they find some estimate like 5 into 10 to the power minus 5 per generation of STRs. And we know that is like many times more and more mutable than the SNV rate, right? So here they use a very, like instead of not using the multiple generation, they use IBD analysis. Like they identify, like genomic regions that are inherited from the common ancestors, right? Shared haplotypes. And then they try to identify what kind of repeat, what is the repeat length probably in the common ancestor, and how many generations are these UK Biobank participants separated from the common ancestor, and what is like how change happened, like how many repeat units changes happened. When you have this at a very large scale across in 100,000, you can really get this mutation rate. And these numbers are really, fascinating, because they when they're focusing on 18 sites, right? Among 18, after filtering, they're focusing on 15 sites, triplet repeat sites. It seems that this is some of the most mutable regions, repeat regions in the human genome. So they estimate like 0.9 per generation. For comparison, like STR genome-wide estimate, mutation rate across the genome is like 5 into 10 to the power minus 5, like, so per generation. And
Wow.
that's like, there's a 2000, these 18, 15 sites are really like 2000 times around
Right.
more mutable than STRs, which are themselves highly mutable. When you compare it with SNV, which is like 2 into 10 to the power minus 8 per generation, right? So you will have like something like 200,000 times more mutable of these 15 sites.
So it's a
That's like,
very small part of the genome, but due to this
yeah.
mutability, its impact on disease is outsized. One of the things must have been very tricky about this analysis, I'm curious to get your take on it, is you have the challenge here of these are, as you mentioned, you're looking at germline expansions. You're also looking at somatic expansions. They're measuring this in the blood. So how do they tease apart the fact that you may be seeing basically expansions in the blood that are showing up as somatic expansions, but how do you determine whether they're somatic or germline? And then you have the related question, which is, we know, for example, because of their previous work in Huntington's disease, that the somatic expansion is happening in the neurons. There's probably things that they and others haven't discovered, because if you're only sequencing the blood, you're going to preferentially pick up things that are somatically expanding in the blood or in the germline, but you're going to miss, for example, most of the time something that is somatically expanding in the liver, for example, but it's not expressed in the blood. So there's I had to stare at this for a long time to try to figure out how they made sense of this, because it's very complicated.
They do discuss a lot of that stuff, and the first thing is like, I think they make sure that these estimates are not really, like affected by somatic expansion, because they kind of validate in different ways, looking at sibling concordance rate, and also actually looking at the data of some repeats expansion estimated in long read data, because we have a a few, some thousand individuals or so for whom long read sequencing have been generated in all of us Biobank. And then also they kind of estimate the repeats based on imputation approach. So basically you don't directly measure the repeat, but you kind of predict it based on the repeats from the other people in the UK Biobank using the shared haplotypes. So they use different ways of validating it. So I think like they kind of made to their best, made sure that these mutation rates are not affected by the somatic, which we will see, in the later part of this discussion. So that's like this one thing that they look at it, and then they also look at the the effect of, for example, they look at both contractions and expansions, right? And so based on the biology of how this repeat expansions happen based on polymerase slippage, we know that this slippage is kind of expansion bias. So you often see expansion than contraction, even though like they are supposed to be stochastic even. So but still there is a bias, and that is what they see here as well. That's kind of confirming what we have seen in previous studies. And they also look at this other interesting, this is like really, I'm very interested in this particular thing, is interruptions, right? So we will, like one of the important thing of studying repeat expansion is what kind of therapeutics you can do to prevent the repeat expansion. So we look at all factors that prevent the expansion. So one is the, like the DNA repair genes, polymorphism, genetic variants and mutations that we have studied in the past, like in the papers where they looked at the, like the age of onset of Huntington's, etc. And the other thing is like, unlike there is like SNV interruptions, like this repeat units are like continuously repeating, right? And that is what is confusing the DNA polymerase. For when there is a mutation, some kind of random mutation, insert a particular SNV in this repeat unit, then it breaks the like the repeating unit, right? So, and that kind of that kind of prevents the expansion. And we have seen this in a lot of spinocerebellar ataxia cases, repeat reports in the past where specific SNVs really are protective, or like protect individuals from expanding across generations. And they were also able to see this in a couple of examples here. They looked at how the SNV interruptions interfere the mutation rate. And they have like a couple of examples. For example, the TCF4 gene, one of the most mutable what they see in this region. So they kind of find that when there is an 18 repeat allele of the TCF4, there's often you see an SNV in the 9th repeat of this expanded, and this kind of, when you have this SNV, it is 135-fold lower odds of expanding further compared to the one without the SNV. So such a large effect of a single SNV on the mutation rate. And similarly for a GLS gene, they look at another repeat, like a particular SNV interruption in the 19 repeat allele that actually decreases the repeat expansion tendency by 3.5-fold. So this is kind of validates what we've seen in, like Mendelian cases where families with particular SNV interruptions, the repeat expansion has been slowed down. This is really super interesting because, we might get therapeutic where at some point, if we are, like the gene editing technologies become safer and able to do in cells, inaccessible cells like brain cells, imagine just, like you really don't have, if you can go and edit the genomic region by just inserting an SNV, you simply can prevent the expansion, the somatic expansion, right? So that's like really super interesting where they look at the germline mutation rate and what kind of interruptions that it prevent or, like increase the mutation rate. So that's been the germline part. So then they actually go to the somatic part. So you had guests talking about, like chip mutations, clonal hematopoiesis, of indeterminate potential mutations, right? So since we started beginning of this exome sequencing era, right? So we've been like, there's been so many papers on chip mutations just for the fact that, like you use blood cells for extracting the DNA. And these blood cells often have like the somatic mutations, which you can capture in using the exome sequencing reads, using this allele fraction stuff, imbalanced allele fraction. And the same concept here, and they use it here to identify the repeat expansion. differentiating between germline repeat and then somatic repeat. And they were able to do so for like 4 genes, I think, with good confidence: TCF4, GLS, DMPK, and also then ATN1. And so they kind of validated different ways. For example, the easiest way to look at the effect, this somatic expansion mutations, is just look at across the age, right? Age is the single most important factor that is associated with somatic repeat expansion. So they show the distribution of, increasing repeat expansion, repeat length alleles as the age increases, both in the UK Biobank and also in all of us. In all of us, particularly in UK Biobank, our age range is very narrow, right? 40 to 60, right? But in UK Biobank, but in all of us, we have a broader age range. So they were able to show in all of us as well. And so the interesting part here, here you can actually identify different repeat lengths, like somatically expanded repeats, and then you can look at the genetics of it, right? So you can able to identify genetic factors associated with repeat expansion, increased protection or increased risk for repeat expansion. And they do this for the TCF4 because the TCF4, like I said, it seems to one of the gene that showed the most unstable repeat, the most instability was seen for TCF4. For example, the statistic was like really interesting. They say like so we know that as the number of repeat increases, the tendency to expand increases.
Yep.
Right. So with the 25 unit, when the TCF4 triplet repeat reaches a length of 25, then by the age of 55, around 1% of blood cells are estimated to be mosaic for the TCF4. And so this is so basically by the middle age, like almost everyone will have somatic, mutations in this TCF4. So this makes it this really interesting gene where you can leverage this fact to create a phenotype of repeat expansion. That's what they did. They created TCF4 repeat expansion, somatic expansion across hundreds of thousands of individuals, right, in UK Biobank. And then correlate the number of repeats, somatic repeat units with the genetics, with the common variants, with the rare variants. And they find really a lot of, genome-wide significant signals. And really the beautiful part here is they kind of compare this with our past reports. For example, we have a GWAS of age at onset for Huntington's disease, right? And this is basically a proxy. There's a GWAS of like a cognitive decline, age of cognitive decline onset. And this is a proxy for, the repeat expansion that's happening in the striatal neurons in the brain.
Yep.
So if you put together there's a Manhattan plot, there is the upper side, you have the TCF4 expansion, and lower side, you have the Huntington's disease. There is there are some loci that perfectly overlaps. It's like a mirror image. That's really amazing to see that, like all of them.
I'm looking at it right now.
DNA
It's figure
mismatched repair genes.
Figure 4, part A, right? It's really interesting. It looks like looking at your reflection in a pond.
Yeah, so it's interesting that some of these loci are really overlaps, right? But what's more interesting is there is heterogeneity. For example, in a couple of MSH2, I think, and there's the other gene, the variant, the common variant, which actually increases the repeat expansion in TCF4, actually does the opposite for HTT in the brain.
And what's the explanation for that? Do they know why it's going opposite directions?
They say there is tissue specific regulation of how this DNA mismatch repair genes handles, this transcriptions. And so they say this is kind of, we expected that, you will expect different effects, heterogeneity across tissue, probably across also traits. And I don't think there's very clear explanation on that, but what is clear is there is a huge heterogeneity.
Yeah.
And there is also heterogeneity in terms of germline versus somatic. For example, TCF4, right? It is kind of seem to be extremely unstable from a somatic perspective compared to germline. But there are genes there is a GLS or a DMPK where they see more germline and very little somatic expansion. And this could be also because these genes, unlike TCF4, which is highly expressed in the blood, the other genes are not that much expressed in the blood. Like transcription is the fundamental part, the driver of repeat expansion. So
Yeah.
the gene has to be expressed in the tissue for it to be expand, right? So that is also another factor. So there are so many differences, but what is interesting is like we are able to, because this is really difficult to do in such a scale for Huntington's disease, like
Right.
diseases that are rare and where you cannot go and find, hundreds of thousands of people. But now you have like endophenotypes or proxies, which you can use to identify these protective associations and stuff like that, right? For example, here, now that sample size is very big, they were able to go and look into the protein coding regions, right? Rare variant associations with this repeat expansion. And they highlight a very nice example where they have like the missense variant in one of the mismatch repair gene actually have a really large effect size on the TCF4 expansion. So now they take this missense variant and go back to the Huntington's summary statistics, look at the effect, and they look at there is really a huge effect of increased accelerated age of onset. And this seems to be a gain of function. Suspect it's a gain of function effect that aligns with our idea of inhibiting MSH3 for, as a treatment for Huntington's. So so this is like really, there's still a lot more data in this stuff, but, I really like this dramatic repeat expansion genetics and also the different statistics they're able to pull out just by looking at a healthy population of the genetics data. And I think like we will see more such data in the near future. And really, this is like a great times for the therapeutics of repeat expansion diseases like Huntington's and other, like spinocerebellar ataxia. And so all these genetic insights that is being generated, day by day will be really useful for this therapeutic development. So this is one topic that I really closely study and, like looking for every day in the literature for all the new findings.
Yeah, I mean, this paper has so much, and this data hasn't been out for very long. I mean, they've had a year probably at most to put this together, right? And it's a remarkable amount that they've done in that time. So kudos to the authors, really. Were there any other final takeaways from that you wanted to share before we move on to the next paper?
I think like, I would like to see, like how this differences in the, like the tissue, like the tissue specific differences, like how the heterogeneity is the reasons, right? So I think it is really important. And one of the important message here is obviously like often people try to use blood sample expansion, repeat expansion data, somatic repeat expansion blood sample to extrapolate the findings to the, the what is happening in the brain, and so it's also tricky and we need to be careful. That's what one of the messages they say here, because we really need to understand the mechanisms in a very specific, in the exact cell type before moving on to designing on, like therapeutics and stuff. And I think that is other major message for me is like to be cautious of, like generalizing the biology across the tissues. There seem to be a lot of heterogeneity in terms of how this modifiers affect the repeat expansion.
Yeah. It's. I'd be interested to have the authors on at some point to see if they have any clever ideas around how you do this across every tissue in the body, besides having hundreds of thousands of single cell sequences across those tissues, or if that's that's what needs to be done to kind of crack this problem. Obviously, that kind of data set would be valuable, not just for repeat expansions, but for many other things. So maybe some government or consortium of governments needs to invest in that. Let's transition to this APOE paper. So this is from a research team that includes Washington University, Denali Therapeutics. We've talked about Alzheimer's and APOE in particular on almost every Roundup. I think it's because it's obviously such an impactful common disease, and the APOE gene is in 10 to 15% of Alzheimer's patients. It's a significant drug target. It's it is about as impactful as it gets from a genetic subtype of common disease perspective. So for that reason, lots of people are interested in figuring out what can it tell us about Alzheimer's biology as a whole, but also is there an opportunity to treat people who carry one or more copies of this gene in some kind of more targeted way? So maybe you could give a quick overview of this paper. I read it in quite a bit of detail this morning, and I thought it was really interesting. I'm interested to hear your thoughts on it, and I'm happy to give mine as well.
Yeah, so I have a very superficial idea about this paper, but it kind of got my interest because of my interest in the APOE and like a lot of recent findings on the APOE. So it is important both from a patient population perspective, right? We know APOE4 is a high risk, like almost recent paper, they show it's like really 100% penetrant at some point of age. And so like any new therapeutics for Alzheimer's, people are trying to focus more on specific population, like recruiting them based on APOE. Then the other side, APOE itself is a target for Alzheimer's, right? And that's like really super interesting area for me. We have discussed a lot of papers there. So, like APOE, targeting APOE could be a therapeutic treatment for Alzheimer's. And we're trying to find supporting evidence, right? So one of the recent paper where we found this sequencing data, they try to identify really extremely rare individuals who carry an APOE4 and then a loss of function variant in the other haplotype that kind of knock down the APOE and how that prevent them from developing Alzheimer's disease. So, and this kind of, like makes it more interesting to understand more about these haplotypes. And so this basically, like APOE has a very important role in the cholesterol metabolism. And that is in the outside the CNS system, right, in the circulation. And we know there is this three haplotype, E2, E3, and E4. And these three haplotypes have different affinities for the LDL receptor. And that kind of results in different lipid profile, right? So basically E2 has, like there is a defective binding of ApoE with the LDL receptor. And so this results in, like decreased clearance of remnants, chylomicrons and VLDL. And also so this causes this, this beta lipoproteinemia, like a Mendelian disease. But at the same time, because of the defective binding of ApoE with the LDL receptor, there is like a increased compensatory upregulation of LDL receptor, and this results in decreased LDL cholesterol. So that happens in the E2. The opposite happens in the E4. E3 is normal binding. E4 is enhanced binding. So enhanced binding results in downregulation of LDL receptor, and that is why ApoE4 individuals have higher LDL cholesterol, right? So, but like this is very well studied, characterized outside the CNS. But the cholesterol, how ApoE4, like handle the cholesterol metabolism in the brain is a bit different, right? So because of the blood-brain barrier. So in the brain, the cholesterol, we do get cholesterol from the outside the CNS that crosses the blood-brain barrier. But the major production happen in the astrocytes, the non-neuronal cell, which produces the cholesterol, and that is also, like it packages with the ApoE, and then it transports it to the neuron. Neurons express LDL receptors, LDLR, but also like other LDL receptors, LRP1. And ApoE's interaction with these LDL receptors is really important, right, to supply cholesterol and other nutrients to the neurons. And this is where, like really we don't know exactly how the different ApoE haplotypes, how their impact on the cholesterol lipid transport, like, results in neurodegeneration. There are a lot of theories about it. The same concept doesn't apply. The same concept in the liver doesn't apply in the, in the neurons. And so that's why this paper was really interesting, because they specifically are looking at how the different haplotypes interact with the LDLR in the neuronal tissues, like they use an in vitro models, but also in vivo models. So ultimately what they show is there is a defective binding of E2 with the LDL receptor. So they show that LDL receptors are expressed in the neurons and also in the lysosomes inside the neurons. And so there is a defective binding of E2 with the LDL receptor. And so this results in decreased intake of, like polyunsaturated fatty acids laden ApoE, like molecules inside the neurons. But in the case of E4, there is like enhanced binding. So there is a increased uptake of these lipids. And what happens is they show there is some kind of recycling defect. So, basically the LDL receptor binds with this lipoprotein and then goes inside the cell, and then they dissociates and the LDL receptor goes back recycled. When there is enhanced binding, they fail to dissociate, and this kind of accumulates the LDL receptor in the lysosomes. So, ultimately what they're saying is this neuron degeneration might be a process of enhanced lipid accumulation. They call this lipofuscinosis in the lysosomes, and this is because of the defective LDLR recycling and the enhanced uptake of, because of the higher binding, enhanced uptake of lipid in the E4. And some protection happens in the E2 where there is a decreased binding. And they also show through experiments that the Christchurch mutation, right, that we know has protective effect on Alzheimer's disease, also have this defective binding and the reduced lipid accumulation in their cells. So it's really interesting in the sense that they kind of show exactly at the APOE receptor binding effect. So this kind of opens up lots of questions like, can LDLR be a target? There have been a lot of studies have looked into the LDLR mutation individuals with familial hypercholesterolemia, or they protected from Alzheimer's disease, or they are at increased risk. I don't think there is pretty good any evidence on that. And also with the PCSK9, I don't know, I think PCSK9 doesn't play that much role in the LDLR recycling in the neurons as like they do in the liver. But again, this opens up questions like, how do you, if you can target LDL receptor, and then prevent this lipid uptake into the neurons to prevent neurodegeneration. Then there is the topic of LDL receptors' other role, right? LDL receptor is very important for uptaking a lot of other stuff that is really important for neuronal health. So maybe you can target LDL receptor and APOE together so that you can be very specific to, interfere only with this interaction. And I think you shared about a particular trial that's happening where they really deliver the APOE2 itself into the brain, right?
Yeah.
So
Yeah, that's right. The only thing I really have to add to this is this therapeutics angle. I think what's interesting to me about this study in general is it shows the multifaceted nature of how genetics is useful in drug development, right? There, you have this allelic series of APOE2, 3, 4 that they showed pretty clearly here has differences in LDLR binding, and they show a hypothesis of a downstream effect on how these end up in the lysis. This tells you a lot of things. It tells you, to your point, if you could figure out how to inhibit specifically APOE4 and LDLR binding with a small molecule, for example, then maybe that would be effective. So that's a therapeutic hypothesis. But the trial that you alluded to is another great example of it. Also might tell you that if you just simply deliver APOE2 to the brain in people who have one or more copies of 4, then you don't necessarily need to know the mechanism, and maybe it still works, right? Because genetics tells you that people who have some APOE2, and I did a little bit of digging to try to understand what the mechanism might be. There's not really a competitive binding. I think some of the lipids would be sequestered into the APOE2, so you just wouldn't get as many lipids going into the lysosome. There's also, maybe potentially some dilution of having a mix of 4s and 2s in there. But there's a trial that read out relatively recently, an early, I think, small phase 1 to 15 people from Lexeo Therapeutics that is using a gene therapy approach to deliver APOE2 into the brain of, I believe, 4 carriers. It's a small study, but I suspect based on the good data they've had so far, they're going to be expanding this. There's companies like Alzion that I mentioned before that are taking a small molecule approach that I don't think follows this mechanism. But the fact that this is a Denali co-authored paper has you imagining whether they might be looking at some kind of gene therapy or small molecule or combo of approaches to go after this mechanism. So it's exciting. I think it's really interesting.
Yeah. You'll know soon, maybe in a few years that, for me, it's a very good point that, you don't need to know the mechanism because genetics tells us really it's protective, right? But knowing the mechanism can take you much further, right? Yeah. In terms of you can be more specific in what you target, not just the APOE2 or whatever, maybe the interaction or specific, right? And that is why it kind of makes very interesting to see this kind of work happening and really people are, dissecting the mechanism. there is like 100 different hypotheses how this APOE, defects causes leads to neurodegeneration, touching up all kinds of biology. And so, yeah, I think it's really interesting to see this evolution as literature. We're getting, more and more clarity, or at least we are learning more on the different haplotypes and what's exactly happening in the neurons.
Yeah, absolutely. Anything else to add on this one, or happy with where we've covered it too?
Yeah, I think it's good. I mean, like we will definitely have more APOE topics coming, and we can revisit this, and maybe our understanding might change a little bit further when we get more. I'm sure that people will start looking at human genetics from the LDL receptor and other pathway genes with this kind of new findings.
Yeah, there aren't really, at least I'm not aware of many LDLR direct associations to Alzheimer's, right? And
No.
I guess the hypothesis, I think there's other ways for APOE to get into the cell besides LDLR, right? So there's a little bit of a redundancy or a buffering there, or are there any other alternative hypotheses to that?
Yeah, that's the main reason. So it's not the only way, right? So there are many other LDL receptors. So it's not really, we wouldn't expect to have a dramatic effect. But also there is, LDL receptor associated myocardial infarction. So people die early, and if they survive to the late life, and that could be because of some other reason. So it's very difficult to study, like the association with Alzheimer's. And I think like people have looked into it, and I don't think there's any striking associations have been seen so far. But, like the gain of function effects or stuff like that, maybe also, sometimes you won't see a protective effect, but you will see the risk effect, like when you really amplify this interaction with some gain of function or something. And these people are at increased risk of Alzheimer's disease, something like phenocopying what they show in this kind of in vivo models, right? So those findings might be also interesting. But yeah, I mean, sure, something will turn out soon.
Yes, yeah, we'll keep tracking it. So we're transitioning back to large population genomics here, but in this case, it's both large population genomics, but also really specific. So this is a decode paper. The title of it is Homozygosity for a Stop Gain Variant in CCDC201 Causes Primary Ovarian Insufficiency. So nearly 175,000 postmenopausal women from Iceland, Denmark, and the UK were included in this study. What was interesting to you about it, and why did you pick it?
So I've been following up on a lot of human genetics work of, this particular trait, aged menopause, and this really picked up after the UK Biobank data. And so for me, this is interesting in different angles. One is from the trait, and the age of menopause that is related to the ovarian function. And like that is one area I'm really super interested in, all the evolving therapeutics for helping women with infertility, and also with like nowadays child birth is like like happening after 40 years, and a lot of women are suffering from it, and we don't really have that many therapeutics, right? So this is a really good area because of a large target population, and human genetics really can play a role, as we have seen in the recent papers. So this really makes it very interesting. And the other thing is the recessive human genetics. That is something of special interest for Decode, also for me, and so they have been doing a lot of work on that. So this comes in that line. So that's also makes it really interesting. So basically the study, what they did is like they did a recessive GWAS. So past GWASs of aged menopause mostly looked at the additive associations. And since the Decode has a special interest in recessive genetics, they looked at the recessive GWAS. They did a recessive GWAS in 175,000 postmenopausal women. And they identified one association, very interesting association, that was driven by a loss of function variant in a gene that's it's only known for like few years now. It's a relatively new gene. Before it was not coded as a protein coding gene. It was thought like a long coding RNA or something. And the effect size is like really huge. It's like almost a decade earlier age at menopause. I don't think even that big of effect size was seen even for a Mendelian causes. Like, for example, the Fragile X mutation, FMR pre-mutation carriers actually one of the biggest problem they have is the kind of ovarian insufficiency. So it's kind of a Mendelian primary ovarian insufficiency. I don't think even there you'd see such a high effect size and penetrance. So that's like really super interesting. So, like I said, there's two things that makes it interesting. One is the biology of the phenotype, and the second is the recessive human genetics. So talking about, like age at menopause, it's a very interesting trait. For the first time, because of large biobanks like UK Biobank, we are able to study this at scale, right? So now when we look at it, we are looking at when we do GWAS and XWAS, we are studying the common variants, rare variant associations. We have learned a lot in the recent years, right? For example, to highlight one of the early, like XWASs of age at menopause really highlighted this breast cancer genes, BRCA1 and 2, and CHEK2. These are very well known breast cancer genes and their effect on the age at menopause. I mean, for the breast cancer gene, at least we know previously that it has an effect on age at menopause because, breast cancer patients often go for, egg freezing before they start chemotherapy. And incidentally, they found that, often they kind of fail to respond to ovarian stimulation. And that is how it led to the discovery that BRCA1 and 2 mutation carriers have primary ovarian insufficiency and they attain menopause really very early age. And so that is known from isolated studies. And now we are kind of, like learning that from a population scale. And the other interesting finding is the CHEK2, which is also, like it's a breast cancer gene, and it is a checkpoint, like regulation. It actually regulates the like the cell checkpoints. And what was found in the UK Biobank was it has the protective association. So basically, individuals who carry CHEK2 mutation have later age at menopause. And this was really interesting from the biology because like basically this, all this boils down to the survival of the oocytes, right? So in case of BRCA1 and 2, so these are all like, like the DNA repair genes, right? So when they are defective, the DNA repair goes unchecked and they go into apoptosis, right? But in the CHEK2, because it's controls the checkpoint, so this is the one that actually, like allows the oocytes to pass through this kind of, like this surveillance. So did these oocytes escape surveillance even though they have defective DNA and they kind of survive, that doesn't go into apoptosis. And this is really interesting because there has been some efforts in, using CHEK2 as a target to specifically targeting CHEK2 in the oocytes to, enhance, prevent, like the oocyte depletion, ovarian reserve depletion in women. So these are like really interesting. And in terms of also, like biology, in terms of Mendelian diseases as well, one of the things that we learned from the UK Biobank like data is that, what we thought was a highly penetrant genetic causes of ovarian insufficiency previously was turned out to be not so much. That's one of the biggest findings. And on the other side, we are identifying new genes that are really highly penetrant, which was not in the current panel for the screen for primary ovarian insufficiency. This is one particular example. The second thing, that's the recessive human genetics, like I said, founder population, Iceland is a founder population like Finland. So they have like special interest on the recessive biology. And, like it's very difficult to study recessive associations because the genotype is very rare. It's like it's like a square of the minor allele frequency, right? So you really don't get that many carriers of recessive mutations in the population to, have enough statistical power to study. And that is one reason why we haven't studied at a population scale. All the recessive diseases that we know is based on Mendelian, isolated cases. Now the sample size is increasing. We're able to look at it, at least for some of the common variants. So Decode has looked at, like systematically, they look at recessive genes. Like, one way to identify recessive diseases is to just, you look at you encounter people with the diseases, then you do genetic diagnosis and you find the genes. But it's often people don't get the genetic diagnosis. The other way is to start from the gene. That's what this Decode is doing, systematically profiling across the genome of all the genes for which there is a depletion of homozygous mutations. What could be the reason? could be early death, or it could be embryonically lethal, or, and they have many examples. And in that line, this is this new example. And so the association, like the biology of this gene is really, interesting. I mean, we don't know anything about the mechanism, but what they see is this gene is actually specifically expressed. The CCTC201 is expressed in the female tissues, the ovary, and also in the uterus, and so they also show a specific expression pattern where they're expressed only in the premenopausal women when they look into the GTEx data, and the expression is completely absent in the postmenopausal women. And when you look at the single cell data, this CCTC201 is expressed in the oocytes. And this is like really super interesting because we are really finding some gene that probably plays a very critical role in the, in the folliculogenesis. And the biology is still, though it's not clear, is kind of hinting at in a very important role. So particularly the recessive associations, I don't know if you came across these findings. There are some growth factors under the TGF beta family, like I think it's BMP15, GDF9. So these are very important for follicle growth, and they have very interesting human genetics. It's also shown in the animals, like homozygous loss, complete loss of these genes actually results in infertility, right? But when there is a heterozygous condition, it actually causes hyperfertility. So it causes twinning, it causes multiple egg activation, and this is actually very well shown in sheep and horses, I think. But also there's like very good evidence in the humans that heterozygous is associated with twinning and multiple childbirths, but homozygous is infertility. And this is because of the very fascinating biology, because these are growth factors which play a role in early follicle recruitment, but also in the follicle activation. And they control how this follicle respond to the follicle stimulating hormone. They act as a brake for their sensitivity. So when they are partially lost, the sensitivity is like the brake is released. So there is a hypersensitivity of FSH, results in multiple follicle activation. But then they're completely lost, the follicle development itself doesn't happen. So, so the same thing is happening here. They have some evidence that heterozygous additive association, there is an increased risk for twinning for CCDC201. There is a homozygous association with the, aged menopause. So it's very well plausible. There is a very similar biology, similar to those, like TGF beta gene growth factors. And that's kind of makes this really interesting. And some estimates are really mind-blowing. Like what they say is like, when you actually look at the premature ovarian insufficiency as a categorical trait, if you put a cutoff of 45 years of age at menopause, the odds ratio is 35. That is 93% of women who are homozygotes have attained menopause before 45 years. And when you look at 40 years, the odds ratio is 27. When you put this in the perspective of how much of this variant will explain the prevalent cases in the population, it seems 0.19% of all primary ovarian insufficiency in the population in the Europe, because this variant is only seen in the European population, can be explained this one gene, this one mutation. That is 1 in every 500 women of primary ovarian insufficiency. And we don't know about this because this was never been in the human annotation before. It's only been recently found. So probably this will go into the gene panel, And
how many genes of this magnitude are on that panel? Are there dozens that have this level of effect?
I mean, there's like more than dozens of genes are in the panel when you actually go for a diagnosis of, infertility and, like primary ovarian insufficiency. And the Nature Medicine paper that I can share it with you from John Perry and others from the, the US, from the MRC Research Institute, they actually showed one of the important findings, like they looked at the penetrance of all this very well known, supposed to be Mendelian genes for primary ovarian insufficiency are not that highly penetrant. There are perfectly healthy carriers in the UK Biobank, right? So it's really this panel has to be revisited. And imagine, so these are the genes that's not there actually. And we can see this, super interesting large effect genes. And that makes us wonder how many such genes are there, like these are one of the traits where recessive genetics plays a very important role. And we are only just beginning to understand the recessive genetics at a population scale because of the limitation of the sample size. Still, we can do so only in populations like Finland and Iceland, where you really have drifted variants, which can, help and find this kind of associations. Yeah, so for these many, many reasons, this is like really super interesting finding, and this kind of touches upon the whole topic of human ovarian biology, and that I've been like really studying in detail recently.
Yeah, absolutely. I think it's an interesting segue into this topic more broadly, which is newborn screening. The fourth and last paper that we wanted to cover is actually one that we covered on this podcast earlier in the year, when I think it was actually the very beginning of the year. I interviewed Wendy Chung for episode 117. If you want to go back and hear about the Guardian program, it's a whole genome-based newborn screening program. Go back and listen to 117 if you want to hear about that in more detail. But that team published a paper just a couple of months ago that described the results from the first 4,000 newborns that were enrolled and screened. And it was a really interesting paper for me for a couple of reasons. First, they published some of the high-level statistics around what percentage of families opted into the program, and also obviously the findings, how many people actually had a positive result. So again, go back and listen to that episode if you want the full breakdown. But as a really high level here, they enrolled about 4,000 families and obviously sequenced the newborns over an 11-month period. 3.7 of those had positive screenings. The majority of those, I think it's important to say, would not have been picked up by the traditional newborn screening. So the heel prick test that goes on as standard in the U.S. national healthcare, or the U.S. federal screening system that we're talking about here. But in most countries, there is some version of this. They focused obviously on a panel of genes, so they are doing whole genome sequencing, but they're only analyzing a couple of hundred genes that have relatively strong clinical actionability off the back of this. So some of the examples of the kinds of diseases that they are reporting on and found are things like long QT syndrome, rare heart disease that can cause sudden infant death, combined immunodeficiencies. There's hundreds of these, but they can be managed often through stem cell transplants, gene therapies, and then also rare diseases like Wilson's disease. So this is I picked this paper because I think it's a large sample size for this kind of program, and it's a good proof of concept, I think, of where this is going. They found that I would make sure I get the numbers right. They found 72% of families opted in to participate. So this is run through six health centers in New York City, and they approached all comers, families who came through these units, and gave them the opportunity to opt in or opt out of being part of this. So they had 72% of people consent to join, and of those that consented, more than 90% also requested the additional screening. They had essentially two tiers of genes. They had the 150 or so genes that they were going to report on in everyone, that these were the most actionable set. And then there was effectively an optional set that included a large number of neurodevelopmental disorders, epilepsies in other cases. I thought that was interesting that a very high fraction of people opted for the expanded panel. We covered some other newborn screening programs previously in the year that I think is important to call out as well, because each of them have a slightly different approach to this. So for example, Early Check is a North Carolina-based screening program that has 3 tiers of panels, including a third one, which is a polygenic risk score for type 1 diabetes. And the reason I think this study is interesting is because I think over the next couple of years, we're going to see these newborn screening programs move quickly into the tens of thousands, and then shortly after that into the hundreds of thousands of individuals. In the UK, for example, Genomics England has started the newborns program here, which has an initial target of 100,000 people. I believe that Guardian and BabySeq and Early Check and a number of others are ramping up in the US. I think these kinds of programs have a couple of interesting implications. One, which is, that I tend to work more closely with therapeutics and clinical trials. You're obviously more steeped in the discovery side. On the therapeutic side, I think there's a huge gap in early matching of patients with rare pediatric diseases to either treatments or clinical trials where there isn't a treatment. And all these programs have been thinking really deeply about how do they make sure, first and foremost, that patients get matched to standard of care, if there is one available, but also working closely with pharma and biotech companies to identify people as early as possible to get clinical trials on the radar for them. And I think we're going to see over the next couple of years, this hopefully have a really positive change on the way that rare and ultra rare disease clinical trials can be run. Because if we live in a world where most babies and most families are offered this, then you could have a really different setup compared to finding that you're starting to have symptoms at 1, 2, 3, 4 years of age, and the molecular problems have been ongoing for that entire time. You can identify it early and potentially have trial designs that involve much earlier treatment that mean you actually have an opportunity to intervene way upstream. So I'm very excited about it from that perspective. I also think they're gonna be really interesting from a discovery perspective as well, because we still have an ascertainment bias challenge in the big cohorts that we're talking about, UK Biobank and others. You mentioned earlier in this episode, the age range is 40 to 65 on enrollment. We know it skews towards healthy. I think we can't expect the ascertainment bias to disappear in these studies either, because people do have the opportunity to opt in or opt out. But I think you're going to get you're going to be able to triangulate between disease cohorts that are ascertained for having the disease, population cohorts, and then newborn or birth cohorts that are at hopefully a similar scale of millions of individuals within the next couple of years. And I think that'll help us understand some of these penetrance estimates with a lot more detail, because for most diseases, it's still kind of tricky. If you just have the cardiomyopathy cohort and the UK Biobank cohort, you don't really know, and you get two very different penetrance estimates from those two cohorts. It can be hard to figure out. So I'm excited from that perspective too, of getting a maybe even more unbiased look at what does genetic variation look like in the population before the forces of selection start to come into play shortly after birth, effectively. What's your thoughts on that? I know you've read it, you've tweeted about it.
So yeah, I mean, I initially misunderstood that it was a rare disease screening, but then someone corrected me actually. Then, so if I understand correctly, this is more of a healthy population, right? Not necessarily that a child has any issues. It's just opting in to know if there's any genetic, conditions.
That's right. Exactly.
But do you think there will be a depletion of rare diseases in the way it is being recruited, or?
Let me think. I don't know that there would be, because these are major. Where I was thinking is if something is detected in pregnancy and people are referred, maybe that would be a source. If they're referred somewhere else, that they're not actually invited to take part in this kind of study, but it's run across, really the largest and most well-equipped centers in New York. So I think they're going to get a really broad swath of the population. And I mean, the numbers also, I meant to mention this earlier, but they had quite amazing stats in terms of the makeup of the population. So they had a very wide representation. I'm just searching for the stats here so I get it right. But it seems like they had a very broad and representative cut of the population. I think there still will be some ascertainment biases. It's very hard to It's very hard to get rid of them.
It'll be very useful in terms of looking at a database for looking at genetic variant frequencies, right? So UK Biobank, all of us are really biased towards adult cohorts, and it's one thing I keep saying, for example, my past colleagues in our university, where we really had a birth cohort that is identified based on this, blood spot samples, just purely unbiased, because you're not really selecting. It's a random selection, and a lot of people die maybe during child or during early life. So you can get a very good estimate of allele frequency. So I think such database is really lacking. Denmark, Scandinavian registry-based biobanks really have a lead to, opportunity to set up that biobank, like UK Biobank, like a genetic database, but also this kind of newborn screening naturally will evolve into a biobank, a database where you can go and look at it. So this kind of tells you, for example, some mutations that result in death during early childhood, early diseases, or even, so that kind of, you can actually identify comparing the allele frequency distribution prevalence with the newborn biobank and adult biobank. And this kind of really helps both in terms of clinical genetics diagnosis as well, right? Often people go and look at one NOMAD or other databases, which is really biased in terms of age and other reasons. And so this is really long time coming. We are badly in need of a database that is representative of very early life children, newborn, And also even before that, pregnant women. That's why I was telling about the embryo samples. A lot of embryo samples in the IVF clinics, let's go wasted, right? And there is a huge opportunity there to establish, tool sequencing and establish a biogenetic database that's super interesting, because you will able to identify mutations that causes, death at the stage of intrauterine death, or even, even before the embryo is formed. And so that end of the spectrum, we really lack. And for that reason, I'm really excited, but also, I guess this will be the future where everyone will have a sequencing done when they're born. It's like do, heel prick tests nowadays to screen for all inborn errors. That will be really easily replaced by whole genome sequencing.
Yeah.
And it seems like there is a very good positive response from the participants. There will be some ascertainment in terms of who is opting in. We will miss out people who lack, who are underrepresented in a lot of socioeconomic factors, but also disease. Some probably depleted of some rare diseases. But that is something that's always the case. We cannot do anything, but the positives are always more than the negatives.
Yeah, and I think we're going to see this conversation evolve over the next couple of years, because my understanding in the US of how it works is, the federal newborn screening heel prick test is very limited, but things do get added to it over time, and it has an extremely high rate of people who take it. I think you can opt out of it, but it's very difficult, and almost nobody does. So I think we will see this trend over time to, if the evidence from these early studies shows that actually this whole genome-based screening it does make sense from a health economic standpoint, then I can see it filtering up into a federal program where you actually may get extremely broad whole genome sequencing with very, very low dropout rate. That opens up some challenging long-term questions, though. We know one of the amazing things about genetics is that you can test once and you have information that's potentially relevant throughout your entire life. We talked about APOE4 status, which will be present in these samples, but of course they're not reporting on it. We talked about primary ovarian insufficiency. Those results would be present in these samples, but they're of course not reporting on it. But we will live in a world where you could potentially hand a child over their genome record when they become an adult at 18 years old, if they choose to, and they can potentially opt in to learn some of these genetic factors at certain points in their lives. I think for good reason, the newborn programs today are staying very far away from this. They're saying we're focusing on pediatric diseases that are clinically actionable, and there's enough work to do there to get that right. But we're going to be confronted with the question at some point, which is, okay, if we do have all this data and we do have a mechanism that you can allow people to get access to it at a certain point, there's lots of other uses for that in prediction and prevention. But, it opens up a huge education and counseling burden as well to try to figure out what we actually do with that information at the right time and in the right way. So I think we're generating a lot of the raw data to potentially power a very different genomically informed healthcare system in the future, but there's a lot of non-scientific things to work out, I would say, to actually see the impact that this is having. But luckily, we've got about 18 years for these kids to solve this problem until they're going to be thinking about this, or 16. I don't know I think in the UK, they'd be turning it over at 16, potentially.
Pretty exciting.
Yeah, it is. It is a lot of work to do. We've got time to wrap up here. Do you have any predictions that you want to make for 2025, or anything that you're excited about that you want to share before we log off?
Yeah, so for me, basically biased opinion. So since I work in this human genetics field, right? So I still feel like human genetics insights into therapeutic development is really very early stages, and we will see more of this, and particularly in diseases for which there has been no good therapeutics before, right? So Huntington's and a lot of other conditions, but also conditions for which people would have never thought about working on a therapeutics, but then human genetics come up and opens up a new biology, right? So I try to hand pick these examples and highlight all the time in the Twitter and the Substack, but I still this is always my prediction in terms of we will see more of these examples. And like I always say, non-coding genome is one of my favorite themes, and whole genome sequencing we did this prediction last year as well itself. And like we predicted, I think we did see a lot of examples based on whole genome data that actually came up published this year. And we would likely see more of it, and because now based on a lot of people are looking at it. But the real challenge would be to how do you interpret the non-coding variants. So that is where I really wanted to see some advancements in how our annotations and the statistical methods, and also like like how we actually study these, make sense of these non-coding variants in when you look at population scale. I'm sure a lot of people are looking at using a lot of creative ways of analyzing this data and identifying this, making new discoveries. So that's something I really closely watch in the next year as well. So whole genome, non-coding genome, and human genetic insights leading to therapeutics, focusing a lot on diseases like, neurodegenerative diseases and very hard to treat diseases, Alzheimer's and
Yeah.
stuff.
Yeah, those are good ones. For me, I'm going to be interested in seeing how the data plays out around what else the GLP-1 agonists can do. We obviously have seen them take the world by storm in obesity in the last couple of years, but there's just amazing data across the board around some of the other things it might be able to do, from Parkinson's to Alzheimer's to addiction. So it is, I don't know, it's kind of blowing my mind how magical these things are. There's also some stories that I'm starting to see come out in mice still at this stage, but around weakening of heart muscle and those kinds of things. So I feel like it will be a year where we will either see some cracks in the facade start to form if they're not as magical as they seem to be, or I think we may see the opposite, which is they just are working in some fundamental way across the board. Perhaps both. I mean, there's very rarely a free lunch, right? In any of these things, things come with side effects. So that's one I'm focused on. I'm feeling like in gene therapies and gene editing, there seems to be like a bifurcation or a tale of two cities right now, where some people still remain incredibly excited around treating diseases that were difficult or impossible to treat before, and you see lots of companies being successful. But on the other hand, it's proving to be very hard, right? Getting these to the brain requires very intensive brain surgeries or other kinds of delivery approaches. That the vectors themselves are having safety problems in many cases. So I think we're at the point right now where some of the hype is starting to meet some of the reality. And I think we're going to see, like always in science, people will find a way, and things will find their way through. But it has felt like this year there's been, for every amazing headline about a transformative gene therapy, there's also been a headline for a major setback where something is just not working as well as people thought, or it's too expensive, or something else. So I'm going to be watching that as well, because I think it's such an important long-term theme. But I think we're going to have some chop in the water for the next couple of years, potentially, as some of these challenges get worked out.
Exciting. Yeah, I'm also interested in those areas as well. So it's really challenging. The recent Rett syndrome trials, one of the patients had some side effect, and really that resulted in a disastrous response from the investors, I think, for the neurogene. And so that's these are examples of how really risky areas, and this kind of make people averse towards taking big risks in trying some like therapeutic ideas. So it's very hard, challenging. We'll see how it goes. I mean, gene therapy has been there for a long time, but never picked up, right? So, and I wonder if gene editing will follow the same path. A lot of people are bullish on the gene editing, but again, there is also the same kind of lot of overlapping challenges that we've seen for the gene therapies, right? So the viral vectors. So it's something to see how things move forward, but the technologies are advancing with no doubt rapidly, right? So I think soon we'll have safer ways of doing this, delivering to tissues like brain and treating diseases that's had no treatment at all before.
That's right. And it seems like ASOs are doing well. Lots is happening there. Maybe there's a kind of middle road here where the gene editing and gene therapies are still scientifically challenging, where the ASOs kind of represent that middle ground where they've been worked on for long enough that some of the major challenges have been picked off, and now we're really getting into seeing the full breadth of what they can do. So we've got a lot to focus on, lots of good stuff ahead. As always, thank you. This is one of my favorite events of the quarter. I know it is for thousands of our listeners. So thank you for taking the time to be here with us.
Thank you very much. Thank you for the opportunity.
We'll see you in 2025. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.